Hello dear, today we are going to discuss about pyrosequencing. The pyros is a Greek word which means fire because the light is produced in this sequence method. That's why we call this method pyrosequencing. In pyrosequencing, determines the sequence of nucleotide by detecting the release of pyrophosphate during nucleotide incorporation in a newly growing DNA strand and monitors DNA synthesis in a real time. As for example, we have a DNA strand, we don't know about the nucleotides pattern present in this DNA fragment and we want to sequence this DNA fragment. We add DNTPs, primer and DNA polymerase and then the DNA synthesis starts. During DNA synthesis, the pyrophosphate is released when incoming nucleotide adds by DNA polymerase and make 3 to 5 phosphodiester bond between thymidine and adenine. This pyrophosphate initiate cascade reaction, which in result the light is produced. This light is detected in pyrosequencing. That's why we call this method pyrosequencing. This pyrosequencing method has been replaced by new advanced sequencing technologies, but this pyrosequencing laid the groundwork for the development of most advanced sequencing technologies like next generation sequencing. Anyhow, and in this video we will discuss about the history or background of pyrosequencing. Many scientists came and contributed by their work in the development of pyrosequencing. Then what is the principle of pyrosequencing, how many steps or process involved in pyrosequencing, how many types of pyrosequencing are used nowadays, what are the advantages, limitations or applications of pyrosequencing, where we can use pyrosequencing. All these elements will be discussed in this lecture one by one. Now we see the history of pyrosequencing. Here is a long list of scientists who came in different era and contributed by their work in the development of pyrosequencing. The first scientist, Pyrosequencing, who came in 1987 and said that the DNA polymerization can be detected by the release of pyrophosphate. As we know, the pyrophosphate is released when incoming nucleotide is added by DNA polymerase. The incoming nucleotide and the already added nucleotide in complementary DNA strand both make 3 to 5 phosphodiester bond and release pyrophosphate, which can be detected in pyrosequencing. Right after 3 years, 1990, Pyrosequencing worked together with Matthias Olin and Betty Patterson. They developed solid phase pyrosequencing where they use magnetic beads and firefly luciferase enzyme for light detection. In 1996, Mustafa Rongi, who introduced modified nucleotide deoxyadenosine alpha-thiotriphosphate. This nucleotide prevents the false signal that usually occurs due to the activity of luciferase enzyme with DATP. So in pyrosequencing, we cannot use DATP because this DNTP is responsible for false signaling. In 1997, the Myren, Olin, Patterson, Rongi worked together with Bajon Astrom. They founded Pyrosequencing AB to commercialize the pyrosequencing technology first time in the history. After this, in 1998, the Ronagi, who introduced a pyrase enzyme which can be used to degrade unincorporated nucleotides. In pyrosequencing, we add a single nucleotide at the same time in many numbers. The few nucleotides are added by DNA polymerase in complementary DNA strand, but the remaining or unbounded or unincorporated nucleotides are degraded by this enzyme. After the discovery of A pyrase enzyme in 1999, the pyrosequencing technology was launched first time commercially. This was done by Pyrosequencing AB. After 6 years, in 2005, the 454 Life Sciences acquired this pyrosequencing technology and developed it into high throughput sequencing technology called 454 sequencing, where they use emulsion PCR. In the end, in 2007, the Roche, the 454 sequencing technology was acquired by Roche. Due to advancement of new sequencing technology, declined the use of 454 sequencing, so that the Roche discontinued to manufacture 454 sequencer in 2013. In this way, the many scientists came and contributed by their work in the successful development of pyrosequencing technology. Now we are going to discuss about principle of pyrosequencing. The pyrosequencing is based on sequencing by synthesis method where we use 300 to 800 base pair DNA size DNA fragment. There are following steps of pyrosequencing. In first step, we immobilize the DNA. We take DNA which we want to be sequenced, and this DNA is called template DNA or target DNA. The newly grown DNA according to the template DNA by DNA polymerase is called complementary DNA. On the other side, we have a solid surface where the magnetic beads like statoviduals are present. When we add this DNA fragment into a reaction mixture, this DNA becomes immobilized when the biotin is attached with magnetic beads, because the magnetic beads has affinity to bind with biotin. In second step, we add primer, which is complementary to the target DNA. This primer provides 3-hydroxyl group for DNA polymerase during DNA synthesis. In third step, we add 4 types of enzymes like DNA polymerase, sulfurylase, luciferase, and apylase. These 4 enzymes are also added into a reaction mixture. And then the substrate of these enzymes like APS and luciferin are also added into a reaction mixture. In fourth step, we add nucleotides one by one into a reaction mixture, as we know that the DNA is made up of 4 types of nucleotide: adenine, guanine, cytosine, and thymidine. Here we use ATPase nucleotide in place of ATP, because this nucleotide prevents fast signaling, which is usually occurred due to the activity of luciferase enzyme in presence of luciferase. So the fast signaling is prevented by this nucleotide. Now we start. How does it work? First of all, we add this nucleotide into a reaction mixture. DNA polymerase add this nucleotide in a complementary DNA strand, and by making 3 to 5 phosphodiester bond between incoming nucleotide and already added nucleotide. When DNA polymerase add this nucleotide and make phosphodiester bond, also release pyrophosphate, alpha and beta phosphate. This pyrophosphate is used by sulfurylase in the presence of adenosine 5'-phosphosulfate, which is used by this enzyme as a substrate and convert it into ATP. And then the next enzyme, luciferase. Luciferase enzyme use ATP in presence of luciferin, which is used by this enzyme as a substrate and convert it into oxyluciferin and pyrophosphate. This enzyme is obtained from American firefly and jack-o'-lantern mushroom. When the oxyluciferin is generated by luciferase enzyme, this oxyluciferin produces light, which is detected and recorded by a sensor called charge couple device. The intensity of this light is recorded in the form of peaks. These peaks are called pyrogram. In pyrogram, here is x-axis and y-axis. The y-axis represents the number of nucleotides which are added by DNA polymerase during DNA synthesis. On x-axis, the sequence of nucleotides. This sequence of nucleotide is similar to the sequence of nucleotide as we add nucleotide into a reaction mixture. In first, we add adenine. Here is a peak of adenine. And in next, we add guanine. Because the adenine is not complementary to the guanine, so there is no pyrophosphate produced. This guanine becomes unincorporated. So there is no peak of guanine is observed in pyrogram. The next nucleotide is cytosine. This cytosine is also not complementary to adenine. That's why there is no pyrophosphate and no enzymatic reaction, and no peak is observed in pyrogram. Cytosine is black. The next nucleotide which we are going to add into a reaction mixture, thymidine. When we add thymidine, thymidine is complementary to adenine. So DNA polymerase add thymidine in a newly growing DNA strand. When thymidine is added by DNA polymerase, again the pyrophosphate is released and whole enzymatic reaction starts. In the end, in a pyrogram, we observe a peak, single peak, which represent to the addition of thymidine. Again, we repeat this step until the sequencing has been completed. So here is the fourth enzyme, apylase, which is used to degrade the unused DMPPs. It means when we add adenine into a reaction mixture, as for example, we add 4 molecules of adenine, and 1 is added by DNA polymerase, and remaining 3 nucleotides are degraded by apylase enzyme. So this apylase enzyme basically clear the reaction mixture from the nucleotides. In this way, we repeat this sequence of nucleotides. We add this nucleotide again and again in a reaction mixture until the sequencing has been completed. And by using this method, we sequence the unknown DNA through pyrosequencing. Now we are going to discuss about process or steps involved in pyrosequencing. How does the pyrosequencing work? There are following steps. The first step is sample preparation, and the second step is PCR amplification, and third step is sequencing reaction, and the fourth step is sequencing analysis. Now we see the sample preparation. First of all, we extract the DNA from biological sample by using mechanical disruption or chemical lysis. After extraction of the DNA from biological specimen, we convert this DNA into small fragment by using an appropriate enzyme like restriction endonuclease. After fragmentation, polishing ends of these fragments is done if unpaired bases are present in this DNA. After polishing, we get the blunt-ended DNA, double-stranded DNA. Now we add two adapters, adapter A and B. These adapters are ligated with blunt-ended DNA by using DNA ligase. As we can see here, the two adapters, adapter B is attached at 5' of our gene of interest, which we want to be sequenced by using pyrosequencing, and the second adapter, adapter A, which is attached at 3'. This adapter, adapter A, is used for attachment of primer during PCR amplification and sequencing reaction, and the second adapter, adapter B, which is used for attachment of biotic. Biotic is attached at 5' adapter. These sequence, because we are going to sequence this DNA, we don't know about the nucleotide sequence in this DNA. So we add these adapter because we know about the nucleotide present in this sequence. So these, according to this sequence, we can prepare primer, and those primers will come and attach. Next step is PCR amplification. In PCR amplification, we use this double-stranded DNA, where the two adapters are present on both sides of this DNA, adapter A and adapter B, adapter A at 3' and adapter B at 5'. So in PCR amplification, first step is denaturation of this double-stranded DNA. Denaturation is done by using sodium hydroxide or by increasing the temperature. This double-stranded DNA is converted into two single-stranded DNA. These two single-stranded DNA are used as a template in PCR amplification. In next step, in PCR amplification, we use biotin tag primer. This biotin tag primer is complementary to the adapter A, which is present at 3' of this single-stranded DNA. As we know, the PCR amplification is based on semi-conservative method. So after one cycle of PCR amplification, the one single-stranded DNA is double-stranded DNA is converted into two double-stranded DNA. So here we can see the two double-stranded DNA are made after one cycle of PCR amplification. This PCR product carry one biotin tagged single-strand DNA, which is complementary to the template strand. In next step, we immobilize this double-stranded DNA, where we are using magnetic beads. These magnetic beads are present on a solid surface in a reaction mixture. These magnetic beads are either like streptovidin or saffrose. These magnetic beads have affinity to bind with biotin. So when we add this PCR product in a reaction mixture, this biotin come and attach with magnetic beads. In this way, this DNA become immobilized in a reaction mixture. In next step, the denaturation. Now we perform denaturation again. In this denaturation method, we use sodium hydroxide. This double-stranded DNA converted into two single-stranded DNA. The one single-stranded DNA where the biotin is present remains in contact with magnetic beads, and the other single-strand DNA leaves the reaction mixture. In next step, we add sequencing primers, which these sequencing primers are again complementary to the adapter A, which is present at 3' of the biotin tagged single-strand DNA. Next step is sequencing reaction. In sequencing reaction, we take template DNA, the template DNA, this biotin tagged single-strand DNA, which is attached with magnetic beads on a solid surface, and the DNA primer is also added at 3' where the adapter A is present. This is used as a template DNA. In a reaction mixture, we add 4 enzymes: polymerase, sulfurylase, luciferase, and apyrase. These 4 enzymes are added into a reaction mixture along with their substrate like APS and luciferase. And then we add nucleotide one by one into a reaction mixture. When we add nucleotide, the next reaction is polymerization. This polymerization is done by DNA polymerase. DNA polymerase start to add nucleotide into a complementary strand according to the template DNA strand. DNA polymerase adds nucleotide according to the template DNA and convert it and produce pyrophosphate, hydrogenase and DNA product. The second reaction is ATP synthesis. This is done by sulforylase. Sulforylase enzyme use pyrophosphate and APS, which is used as a substrate by this enzyme, and convert it into ATP and sulfatide, SO4-2. The third one is light production. This ATP is used by luciferase enzyme in presence of luciferin and oxygen and convert it into AMP, pyrophosphate, oxyluciferin and carbon dioxide. This oxyluciferin produces light which is detected and recorded by a sensor. Then the next one is degradation. In degradation, a pyrase enzyme convert unused DNTPs or unincorporated DNTPs, those nucleotides which are not added by DNA polymerase in a complementary DNA strand. These unused DNTPs are converted into nucleoside and phosphate by a pyrase enzyme. So in this way, the reaction mixture becomes clear from unincorporated nucleotides. The next one is light sensor. In pyrosequencing, we use charge coupled device, which is this device detect and record the light which is produced by oxyluciferin in the form of peaks. The last step of pyrosequencing is sequencing analysis. In sequencing analysis, the intensity of emitted light is detected by using charge coupled device and recorded in the form of peaks. These peaks are called pyrogram. So in sequencing analysis, we generate a pyrogram. Here we can see a diagram of pyrogram. In pyrogram, on x-axis, the sequence of nucleotide, which is according to the nucleotides added into a reaction mixture, and on y-axis, the number of nucleotides which are added by DNA polymerase in a complementary DNA strand. So here we can see the first nucleotide is adenine. This adenine show a peak, it means that the adenine is added by DNA polymerase in a complementary strand and then release a pyrophosphate. This pyrophosphate initiated a enzymatic cascade reaction and then the light is produced. So this light is detected and recorded by a sensor in the form of peak. The next nucleotide is guanine. If there is no peak, it means this nucleotide is not added by DNA polymerase in a complementary DNA strand. The next nucleotide is cytosine. Here we can see the intensity of light is increased because the double nucleotides are added by DNA polymerase. The peak of this nucleotide is also double as compared to the adenine where the single nucleotide is added. The next, thymidine and adenine, both nucleotides are not added by DNA polymerase, that's why there is no peak of thymidine and adenine. The next nucleotide is guanine. Here is a single peak of this nucleotide. In this way we can read this pyrogram. Now we make a DNA fragment. The first nucleotide is adenine, the second nucleotide is cytosine, third nucleotide is also cytosine because the peak of this cytosine is double. It means there are two nucleotides of this of cytosine. The next nucleotide is guanine. So in this way 5'3" and we make a DNA fragment. This fragment is complementary to the template DNA. In this way we perform a DNA sequencing by using pyrosequencing. The multiple fragments are sequenced by using pyrosequencing and then assembled into a complete genome sequencing by using bioinformatic tool, which can be used to sequence the DNA. The first type of pyrosequencing is solid phase pyrosequencing. As the name indicates that there is a solid surface where the magnetic beads are attached. These magnetic beads are used to immobilize a single strand protein tag single strand DNA. This protein has affinity to bind with magnetic beads, so in this way this DNA becomes immobilized. In a reaction mixture, we add three enzymes like DNA polymerase, sulforylase and luciferin along with their substrate like luciferin and APS. In this method, we perform a watching step that because there is no apyrase enzyme, so we perform this step to eliminate the unincorporated DNTPs from a reaction mixture. This method was the original method that was developed in the late 1990s. This method provided the foundation for the development of other sequencing methods and even played a vital role in the development of next generation sequencing method. Second type of pyrosequencing is liquid phase pyrosequencing. as the name liquid phase indicates that the sequencing is performed in a solution or in a liquid phase. In this method, single-strand DNA is prepared and hybridized with sequencing primer in a solution or in a liquid phase. After single-strand DNA preparation and hybridization with primer, we add nucleotide in a liquid phase and then pyrophosphate is released, which is converted into ATP and then detected as a light signal. This method is less commonly used in high throughput application but simplifies the process and reduces the complexity which are associated with solid phase pyrosequencing, because there is no use of magnetic beads and biotin as we use in a solid phase pyrosequencing. In this way, it reduces the complexity and simplifies the process of DNA sequencing. Third type of pyrosequencing is 454 sequencing. First successfully commercialized NGS technology was developed by 454 Life Sciences that combines emergent PCR with pyrosequencing. In this method, the DNA fragments are amplified within teeny droplets of oil in water emulsion. As we can see here, the water in oil emulsion, this droplet carry the beads on which the primers are attached. The single-strand DNA, which is used as a target DNA or which we want to be sequenced by using 454 sequencing, this single-strand DNA hybridized with primers on a bead. So in this droplet, the DNTPs and polymerase enzyme are also present. So in 454 sequencing, first of all, we isolated the DNA. This DNA is fragmentized by using restriction endonuclease. This DNA is converted into small DNA fragments. These small DNA fragments are amplified by emergent PCR. After amplification of this DNA, then we perform sequencing by synthesis method. Each beads in a emulsion carry single template DNA which we want to be sequenced by using 454 sequencing. Now we see the advantages of pyrosequencing. The first advantage of this method is this method provides real-time sequencing data that allows monitoring of sequencing process as it occurs. The second advantage is this method is high throughput method in which we can process multiple samples simultaneously, which is useful for large-scale genome sequencing. The third one is the results are directly converted into pyrogram. There is no need of post-sequencing electrophoresis, which reduces the cost and time. The fourth is this method having fewer steps for sample preparation and pre-processing steps as compared to other methods. The last advantage of this method is there is no need of radioactively labeled nucleotide as we use in Sanger method or chemical sequencing method. So in this method we use natural DNTPs. So in this way it reduces the cost and make the process simplify. Limitations of pyrosequencing are given below. Here the first one is pyrosequencing produces short read length as compared to others. That's why we cannot sequence the large genome, large DNA fragments and the fragments carrying repetitive nucleotides. The second is the pyrosequencing cannot detect accurately long homopolymers or the sequences that contain repeated nucleotides, in result which leads to sequencing error. The third one is although the pyrosequencing is less expensive than some NGS methods, but the cost per base pair is higher than other methods. The fourth one is the pyrosequencing has been largely replaced with more advanced sequencing methods that offers a longer read length and high throughput at low cost. So these are the limitations of pyrosequencing.